Thank you, Janet, and thank ARM for hosting what turns out to be an exceptional event year in and year out. And thank you to the audience as well for attending this Fireside Chat during the Cell and Gene Therapy Investor Day. So I'm Gbola Amusa, as Janet mentioned. I'm a partner director of research at Chardan, where our vision is to really identify companies with the potential to create real value for society and therefore see exceptional investment returns. And so disruptive innovation is what we're about, and I'm really pleased to be here with one of the disruptive innovators over time, Dr. Mark Kaye, a true luminary who heads the Kaye Lab at Stanford. He focuses on gene therapies and small RNAs for the treatment of genetic and acquired diseases. So thank you, Mark, for joining.
Thanks, Gbola, for inviting me. And
Great. I'm not sure that everyone appreciates your contributions to the field of gene therapy, so would you start by giving a little bit of an overview of your research accomplishments and your areas of focus over time?
Thanks. Yeah, so I actually got interested in gene therapy when I was doing an MD Ph.D. And in my last experiment before going back onto the ward, I was using a new technology called P-element transformation to make transgenic flies to try to prove a genetic study in flies. And while I was doing all this work, I kept saying, wow, this will be great to do it in people. And then I started thinking of all the years it would take me to finish, going back to medical school, residency, fellowship, and I thought by that time all the interesting diseases would already be treated because the concept seemed so simple. But obviously that didn't happen. And I started my career in the field in 1990 as a postdoc, and over the years I've worked on many different vector systems. In the early days, we were excited by just transducing just a very few cells in a tissue. And over the years, as the vector technology got better, I started to collaborate with a group at Somatics on AAV vectors. And we actually were then published the first study showing that AAV could be administered by simple intravenous infusion into a mouse, and that we could get curative factor IX levels that persisted. And unlike the other vectors, there was no toxicity. And this was really the aha moment. And ultimately, I had the original IND. It was a collaboration between myself, Kathy High, and a company called Avogen, where we went into the first trial, phase 1/2 trial, where AAV2 was administered intravenously for human factor IX, subsequently the T cell response, etc. So over the years, my lab has focused on a lot of vectorology. We work a lot on mechanistic insights that help us improve the vector systems. Most recently, we've been focusing a lot on using capsid biology, shuffling and evolutionary selection technologies to select for AAVs that have improved properties. We've developed an AAV non-nuclease-mediated homologous recombination approach for permanent correction, even something that could be used in neonates. And then the other part of my lab works on non-coding RNA biology, with the idea that as we discover the function of non-coding RNAs, we will be able to intervene for certain types of diseases. And this all really started by trying to merge RNAi therapeutics with vectorology and getting results that were unexpected and following up on those results. So that basically summarizes a lot of my scientific career and interests.
Great. Could you give us your version of a history of gene therapy, let's say since 1990? What were the major challenges and what actually got us here to the point where we're seeing all these acquisitions in AAV-based gene therapy and also previously in ex vivo gene-modified cell therapy?
Yeah, so I think in the very early days, when people started to find vectors and they could do gene transduction, there was a huge wave of excitement, and probably the excitement was a bit overblown because people were starting to promise, like in 5 years we would treat this disease or that disease. And yet as a young person in the field, I realized there were a lot of technical barriers that there was no obvious way to get around at that time that was really making it reasonable to say 5 years. It was possible that some of these technological barriers would be overcome within 5 years, but it was unclear. So I was a little bit concerned about the hype of the field at that time. A lot of philosophy or thoughts were, well, we'll start treating patients even if it's not going to be therapeutic so we can learn something. And then things like Jesse Gelsinger happened and kind of set the field back. A lot of scientists and physician scientists started to think of gene therapy as a pseudoscience. I was told this many times, and so that's where things were. Now, when AAV came on the scene, there was a lot of potential excitement about it. I think the original vector described by Jude Smolsky, the idea was that it would integrate in this certain locus and this and that, which really doesn't turn out to be the value of AAV. But at the time, that's what it was thought, and I think it was a visionary type of approach. But when you went to the gene therapy meetings or meetings where there was discussions about gene therapy, it was hard to even make enough AAV to inject a mouse, let alone humans. So I know that there are bottlenecks now in manufacturing, whatever, but we're talking about on orders of magnitude different scale as to what the issues were back many years ago.
Great. And so what's currently going on in research in companies or academia that currently excites you, whether it's in your lab or outside?
Well, I think it's great that there's a lot of, from the more of the company or the clinical side, I think it's really exciting that many of these technologies are now being tested in human clinical trials. I think, there's a lot of disorders that people think of as low-hanging fruit, but even those, like when we did the original hemophilia trial, we looked at every possible thing that might happen. But this immune response, if you will, that leads to sometimes the loss of the transduced cells, this was totally unanticipated based on all the preclinical studies that were done. So I think that it's really important when you have a sound scientific basis to go forward into the clinic and start treating patients, because until you treat a human, you just don't know what's going to happen. But if you step backwards, I think if you look at what's coming along the line, there's a lot of excitement in the field of genome editing or gene therapy combined with genome editing. And sometimes I think people like to categorize these things into different buckets, but they're really overlapping continuum. And when we talk about genome engineering, it's not all one and the same. There are many different forms of genome engineering. Some of these are going to be used very soon in clinical trials. There are other forms that may be a few years down the line. And if you look in laboratories, I think some of the exciting things are single nucleotide base editing technologies. There's a lot of interest in doing this type of work. I think that some of the work that I'm most excited about, and to mention that I am a co-founder of Logic Biotherapeutics, and a lot of this has been licensed to them, is the ability to use non-nuclease mediated approaches with AAV to basically add a gene sequence onto an existing gene, so we don't disrupt it, but we just add it on, so it takes on the gene expression profiles that the endogenous gene does without interfering with the normal function. We call this gene ride now. So I think that this is early on, but it's ready to be tested in the clinic. And I think there's a lot of interest in approaches that could increase efficiency and use in other applications. And finally, I just want to say that I think that the work, the human genome work, is extremely exciting. And although it's ancillary to gene therapy per se, when we understand what the genome does, and the majority of the genome doesn't encode protein sequences, but encode the dark genome or non-coding RNAs, if they're called, we're really just trying to understand what these things do now. And as we understand their function, I think there's going to be a lot of room to manipulate them. And as the gene transfer technologies get better, and as we understand what these do, I think it's going to open up whole other pathways to treatment of other types of diseases.
Just a quick follow-up. You mentioned non-nuclease-based approaches in genome editing. What problem were you trying to solve when you did work in that area?
Right. So one of the issues, I think, for in vivo gene therapy is that if you are giving a nuclease in vivo, there's always a potential for off-targeting, things along those lines. I think that once people can limit the expression of the nuclease, it becomes a much safer technology. But as a pediatrician, I'm very interested. In fact, I went into gene therapy to treat inborn errors of metabolism because that was my clinical specialty. And it's clear that with vectors like AAV, if you treat a newborn or a young individual, as the tissue or organ grows, you lose the transgene expression. And as we may talk about later, it's difficult to re-administer. So if we could do something where we could get the gene to insert, it's been long known from lots of studies that once you have integration into hepatocytes, you'll get lifelong expression. So one, we wanted to be able to do single-shot therapy to basically treat a disease so we don't have to worry about re-administration. Two, we didn't want to worry about the complexities of adding a second product, which is a nuclease. And I'm not against that approach. I'm just saying it makes it less complex. And there are some concerns based on animal studies that giving high doses of AAV in infancy, at least in animals, can result in liver tumors. And this has always been shown in mice to be due to integration at some rare event where the promoter from the vector inserts next to a proto-oncogene and activates it. So the vectors that are used in this gene-write technology, there is no promoter in the vector. So even if you do get some off-target integration, you in theory now diminish the risk of things like hepatocellular carcinoma.
Great. So for a while in vivo gene therapy, the liver, the eye, the brain were the primary target tissues. And the thing I like talking to academics is sometimes I hear about the future. And a year ago, some were saying muscle might emerge. What do you think is the next primary target tissue to potentially emerge?
Well, from an in vivo point of view, I think there's been outstanding results in the muscle. I mean, I think the data recently with some of the DMD trials is much better than I might have anticipated. It depends what you call about SMA. It's really a neurologic disorder. I think the data there looks outstanding as well. So I can't remember if you said we're including CNS in already. I think CNS is a target tissue for which there's a lot going on, and there's a lot of potential to treat some pretty devastating disorders. I think the heart, for which there have been some failed trials in the past, probably because the best AAV wasn't used in those cases. I think you're going to see a resurgence in interest in using gene transfer to treat heart disease. There's a number of both academic and corporate entities, or commercial entities, I should say, working on ways to transduce the heart. So I think, when I think of the heart, I think in some ways it's great. It's the least mitotic tissue in the human body, and that's why you don't see heart tumors very often. But I think the thing that is interesting about the heart is that there are a lot of people trying to treat some congenital heart defects or genetic heart defects, which are individually rare. But then there are the more garden variety heart defects or heart problems that develop later in life that people my age start to think about. And I think there isn't as much effort in this as I would think, but just from my own thoughts. And part of it is to get drugs approved for things like heart failure, myocardial infarction, requires treatment of quite a number of patients, so those trials get extremely expensive. So I think heart and CNS for in vivo. The other area to keep in mind is the pancreas. There's a lot of efforts now, and there's a lot of data about you can use two genes that are relatively small, that if you put them into the alpha cells of the islets, they can get converted into beta cells, and both in human islets as well as in mouse, and it's been used to treat diabetes. But there'll be other complications possibly down the road, but I think this is a target that you're going to see people go after very soon as well.
Could you say a little more on CNS, and let's exclude neuromuscular from CNS?
Sure.
What else might be lower hanging fruit for gene therapy in CNS?
Well, I think there's a lot of interest in some of the neurodegenerative disorders where the mechanisms are being more well worked out due to repeat expansions versus production of small peptides or abnormal production of RNAs. There are ways to try to deal with that, but I think the more common variety type disorders that many of us also probably in our age think about are things like Alzheimer's disease, for example. And I think that although there's still some debate about the mechanism of Alzheimer's, there are some really valid and potential gene targets there. For example, the ApoE4 allele is one example. Tau is another target, and there are clever ways to put in genes that will act as antibodies or binding to stop the tau from accumulating, or changing somebody who's ApoE4 homozygous to ApoE2, what have you. And whether or not this is going to work, I think is not clear, but there's enough scientific principles that I think it's a really valid approach.
Great. Let's talk vectorology a little bit. The current paradigm oftentimes seems to be AAV for in vivo purposes, lentivirus for ex vivo purposes. Do you see scope for other vectors to figure in driving innovation going forward, or even for, let's say, lenti to start to be used for in vivo applications?
Yeah, I mean, I think this is a good point and question. You know, we also work on non-viral vectors, but we've worked on it from the DNA side, making DNA molecules that are more robust at expression because they're not transcriptionally inactivated, which is another mechanism that's been learned over the last few years. But if we look just in the non-viral vector space, people have not been successful at delivering DNA in vivo in therapeutic levels that are safe. The RNAi field came along after people had been working on DNA, and a lot of people like myself were skeptical that that would work. But the RNAi, the chemistries are different, the molecules are smaller, and you don't need to get nuclear import to get these things to work. And as a result of that, there's a huge excitement about RNAi, and there should be. There's an approved product, and there's lots of possibilities there. But then people start to say, well, if we can do it for RNA, why can't we do it for DNA? And I try to remind people have been working on DNA for years before RNA. But maybe now there's a resurgence, and there'll be something that comes down the line that will be a new, totally new modality. But there's nothing that I know of that's right on the horizon that I think, okay, this is going to be a potential slam dunk. If you talk about other viral vectors, and I think it's important to talk about lenti vectors, we published early on about in vivo lenti transduction in mouse models. With the improvements that have been made, there's a lot of interest now in using lenti in vivo in the liver, because you do get integration, so you wouldn't have to worry about loss of expression. I think the concerns there are, number one is I think because it's an envelope virus giving high doses, there could still be some toxicity issues. And secondly, by definition, a lentivirus has to integrate. And I think insertional mutagenesis issues are still something that people should be concerned about. There are non-integrating lentises, but the transgene expression from those at this point, the expression levels are fairly low. So I also have some skepticism unless there's major improvements about how well that will work. And I know there are other groups now working on other viruses for which we know very little about, but potentially could be new vectors in the future. I think that over the years, you hear about the vectors that you've mentioned and you all know about, but there have been a number of other viruses that people have tried to exploit over the years for gene therapy. Herpes virus, for example, and there's some alpha viral vectors. There's all sorts of other viruses you don't know that much about because people really didn't get them to the same level of sophistication that they could be used for in vivo gene therapy.
Great. So an investment opportunity question. We know that limitations of breakthrough technologies create further investment opportunities. So can you talk about the limitations of AAV-based gene therapy and what innovations maybe are needed to sort of actualize the full potential of the tech?
This is a good point. And I want to make one point that I think is important. I don't think there's ever going to be one vector that's going to supplant all vectors. It's one size will never fit all. That's my prediction. So I think you're going to see different vectors for different opportunities, and there'll probably be some changing of predominant vectors at some level over time. But the AAV isn't perfect, okay? It's far from perfect. So the limitations are the genome size that what we can package, limited to about 5 kb. The second is pre-existing immunity, which precludes individuals from getting the AAV. If it's a systemic delivery, again, depending on what you're using it for, this can be a bigger problem than in other situations. And I think that it's not unreasonable to think that there may be several serotypes of AAV for every disease. And you get checked and you get the injection of the one that you have least pre-existing immunity to. The other part of the immune response is the ability to re-administer. There are clever approaches that people are trying to use so that if you're not pre-immune, that when you get the first dose of AAV, what actually happens is that you don't develop this humoral immune response. I mean, all of these have risks associated with them, and I think we just have to, as a community, have to continue to do the science to see how useful this approach will be. The third limitation is the presumed T-cell mediated response. I mean, the exact mechanism, there's still some debate about that, but you can get, for example, in the liver, you can get transaminitis, which is thought to be T-cell mediated, although it hasn't been absolutely proven, that is directed against the capsid itself and eliminates the genes, the cells that have taken up the vector. And this hasn't been a showstopper, but it has caused some issues, and it's a limitation. And then I would say finally is the inability to get AAV to go into certain tissues and to persist. And again, there's ways to try to circumvent that, but it is a limitation at this point in time.
So do you see a lot of room for capsid engineering to solve some of those issues, or is capsid engineering really just to get around IP?
Yes. Well, I don't think it's to just get around IP. I mean, we do a lot of capsid engineering, and honestly, as an academic, I just want the best thing that's going to work. Honestly, the community has split views on this. Some people in the community say, well, we have enough AAVs out there. We just need to learn how to manufacture those the best, deliver those the best. And there are others who say, well, there's a lot existing in nature. We just have to use those and figure out which one. I think figuring out which one is a huge scientific field and of great importance. And then there's a third group who says, well, let's do nature in high speed and let's make all sorts of variant AAVs. but then we have to figure out ways to put them through artificial evolutionary selection for a particular property. And I think there's huge room for improvement on AAVs using this approach. There's lots of different variations on that theme. People do it different ways. I don't think there's one right or wrong way to do it, because we just don't know. You can select for things that, and we, if we want, we can talk a little more about some of the ways that people do selection and why they would do it a way they do. I mean, for example, you can try to select for something where there's little pre-existing immunity in the human population. And the question I have is, if you really select for something that's zero, meaning that nobody has pre-existing immunity for, will it function in humans? Maybe you could, those could be two properties that if you really get to where you want, the two never coordinate with each other. But I think time will tell about this. Clearly, there are AAVs for which there's less pre-existing immunity. There's a lot of confusion in the field because people measure this very differently. They use different patient populations. So it's sometimes very difficult to know what the true answer is in terms of things like pre-existing immunity.
Great. So when we hear about a new capsid and it has some promise, are the right dimensions on which we should be evaluating it? Would it be protein production as one, and tropism maybe we can include in that? Number two would be, let's say, immunogenicity, and number three would be manufacturing yields. Would you add any other dimensions?
Yeah, I think species issues, because you can find capsids that work well in mouse or maybe even non-human primates, but don't necessarily translate into humans. And honestly, I think that's one of the biggest challenges for the field, is which animal model do you use to select for your AAV, and that's a topic that I'm very interested in. And there's not one perfect animal model by far, but the other points that you make is, I think, are very valid points. Immunogenicity issues, manufacturing, I think, is something that has to be taken into the equation. And if there is a trade-off for one property versus another, where do the lines intersect, and what would make you happy as a commercial entity to bring this product forward? And it may be dependent on what you're trying to treat, what tissue you're trying to deliver, because the amount that you need to manufacture for systemic delivery in an adult versus intraretinal or intraocular administration is extremely different.
Sure. And so with AAV-based and other types of gene therapy, we've seen some tremendous valuation inflection points for companies who hit on phase 1-2 data. So I think that ups the stake potentially for preclinical data sets. So can you talk about the right models? We don't have much time sometimes to evaluate a product getting results soon. So what preclinical models should we focus on? What's a good way to frame data sets?
Well, I think this is, again, not one size fits all. It depends on what tissue you're trying to target. And what's known about, so first of all, is there a disease animal model that recapitulates the human model? Because at least initially showing efficacy in there can be very helpful. There are a number of animal models, a lot of times rodents, where the gene defect is present, but the phenotype can be different than what's seen in humans. So that adds a level of complexity to the analysis. But there are things like, if you want to look at efficiency of transduction, there's huge variation between species, and there's really no easy way to predict this. So I think you have to use sometimes a combination of preclinical models. So some people say, okay, you start in the mouse because those are easiest and the least expensive to work with. But then you have things like humanized mouse models, where, for example, in the liver, there are ways to trick the liver so that you can inject primary human hepatocytes and repopulate the mouse liver with human cells. So you can look at transduction efficiency in an in vivo human setting. The difference there is these are immunodeficient mice. All the other cells of the liver are mouse, not human. So there are limitations there with this model, but there's some advantages to this model. People are developing human three-dimensional organoids and how this might be used for prediction. And then there are other large animal models besides the non-human primate, like some dogs, because dogs have some of their disease models for some of these things in dogs. And what combination you use, I think, really has to be set up based on the disease you're trying to treat. What's known about the AAV capsid? Has it been used in humans for the particular tissue target? And if the target is, and we know like what the transduction efficiency is, what the likelihood of a T cell response is, what the manufacturing profile might be. If it's a brand new capsid, you have to start a little bit back and do a little bit more. But as time goes on, I think we're going to learn these things.
And so, can you talk a little bit about the potential for tox issues with cell and gene therapies, and how should we frame that? How should we investigate whether a product is going to be good or less robust?
Yeah, this is another important issue. You know, all the commercial entities, there's lots of variations in the manufacturing that's being done. There are mammalian-based, insect cell-based, and even within those, there's lots of variations. Even if it's the same serotype, after manufacturing is done, the post-manufacturing or the post-production purification schemes are different. There's differences in empty capsids, for example, versus full capsids, versus this or that contaminant. We honestly don't know. I mean, we use DNA titer as a way to measure like vector genomes, but you can make the same lot by the same process, and then you test for potency in some model, whether it's cell culture or animals, and there can be a big variation. But how would that translate into different potency in a human? We don't always know. So the standards for that are lacking, and it's not due to anybody's fault really, because we just don't know which parameters are most important. And I think all of these things can influence potential efficacy as well as toxicity. I think this, presumed T cell response, or this T cell response that we see, the problem is there's no good animal model to study that in. So we can't do the types of basic things we would like to do. We were at another conference, and there was some discussion about actually biopsying now the patient's livers when they have this LFT elevation, so we can study in more detail what's going on. And there was debate amongst people whether they thought that would be acceptable or not. But these are things I think that have to be discussed.
Great. Let's talk about the future a little bit. We've heard a lot, or increasingly, about synthetic biology. So can you tell us what that is and why it's particularly relevant for gene therapy?
Well, synthetic biology really is such a general term. It's hard to always frame it. You can talk about synthetic biology, do promoter bashing to make better promoters. You can talk about synthetic biology even with these artificial capsids. But I think what you're also going to start to see are that people are going to modify the capsids chemically or after their production by adding different ligands on. I mean, these kinds of things have been tried with viral vectors in the past, and they haven't worked really well. But I think there's some very new approaches that people are trying, and maybe they'll be successful. So in that way, that's also a form of synthetic biology. I think, too, you can talk about making new protein coding sequences that maybe don't exist exactly that way in nature, have some new property, like CAR T cells as an example, even though it's ex vivo. I mean, these types of things, all of these can be, I think, considered synthetic biology. And finally, we've been trying to do what viruses have found ways to do over millions of years, is to make nanoparticles that can deliver DNA through synthetic biology. and maybe there will be some breakthroughs in that area that will then bring out this whole new field.
Great. So how far are we from treating mass market diseases using in vivo gene therapy? I think the low-hanging fruit from our perspective has been monogenic diseases, clear phenotype, small amount of protein is transformative, tropic vector, right delivery.
Well, let me just say, when I started in the field, nobody, the only groups that were interested in monogenic rare disorders was the NIH. Couldn't get any commercial entities. And I remember going to some of these commercial entities, some of who you know, and pleading with them. And at the time, there was no interest. I mean, obviously, things have changed a lot. But I think we're going to start to see, we already are starting to see trials for these mass market types of disorders. And at what level do you call it mass market? But the examples I brought up before, for example, Alzheimer's disease, I think that would be considered a mass market. I mean, there's trials for Parkinson's. Maybe that's not, it's acquired, semi-mass market. I think you're going to start seeing trials for things like diabetes. And again, I think there's going to be a resurgence in interest in treating things like heart disease. And then finally, one other thing we didn't touch upon, but there's been a lot of advancement in the ability to make antibodies and to understand like pan-neutralizing antibodies against serious infectious agents like HIV or influenza. And if we can express those from a tissue, we may be able to use passive immunity approaches using a combination of gene therapy, genome editing, what have you, to protect people from these types of infections, which I also would call kind of a massive market strategy.
Great. I'm going to ask you a question that you should perhaps be asking me. And we're post-Kite, Juno, Avexis, Spark, Nightstar being acquired. But from your perspective as a scientist, what's driving this wave of M&A?
Well, I think it really is showing that technology is validated now in humans. And although we are seeing success and what have you, I think now it really validates the fact that when the big pharma has really taken such an interest in this, that it validates the approach. And I think it shows insight that there's much more that we can treat as the technology matures. So that's very exciting for somebody who's been in the field since 1990, when there was very little interest from commercial entities to pursue this. So I think that's it's exciting for even us that work in the laboratory.
Great. And then on the instances where you speak with investors, what do you think they're missing most about the science side of cell and gene therapy?
Well, it's a little bit of a difficult question because there's different
Knowledge
levels
goes-
of knowledge
right.
Amongst the investors. I mean, I've had meetings and times where we sit down and we talk about like which capsid for a particular organ, and I think there's some frustration after an investor say, well, can't you pick the exact serotype to tell me which one we should pursue? And I said, well, if it was that simple, we wouldn't be sitting here right now discussing it. But I think sometimes there's just, and it depends, and sometimes it's not the exact same topic, but there's sometimes a fundamental misunderstanding of how vectors work, and the fact that these are macromolecular structures, and sometimes we can't get them to other tissues, not because the cells don't have the receptors, but because the physical barrier of the way the vasculature basically brings nutrients into that tissue. So a lot of times after we've talked about this, they say, I didn't realize this or that. So I think it's just like anything, just a matter of education.
Great. And last one for me, and maybe there's time for some audience questions, but what do you think we'll be talking about, the audience, everyone here, at the ARM Cell and Gene Therapy Investor Day 2020 and let's say 2025?
Well, I think you're going to see a lot more of these low-hanging fruit disorders treated, products more, there'll be a trickle more products. I think we probably are going to have some setbacks in the field. I just hope they're not real serious ones, but I think people should be ready that this may happen. But I think what you're going to see and what's going to be really exciting is the innovation in the science, advances in vectorology, advances in understanding how the vector and the host interact with each other to elicit these responses, and perhaps novel approaches to get around this immune response, some of these immune response issues. So I think in the 5-year period you'll see that. I think in the 10-year frame, you're going to see things like nucleotide base pair editing type things really starting to take more fruition. And I think people have to really be careful right now and realize that gene therapy, genome editing, there's a lot of overlap. They're not all the same. Some are really, in full swing, at least for the early trials. But there are going to be more complex ways to do genome editing in the future to do more sophisticated things. And I think these are the things you'll start hearing about in the next 5 to 10 years.
Great. And I think we have time for questions. Anyone? Great. I'd like to thank Dr. Marquet.
Thank you.
Thank you.